Hello, everyone. Welcome to today's live broadcast, Good Plastics, Bad Plastics, How Tubes, Plates, and Pipette Tips Can Affect Your Experiments. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Eppendorf. Eppendorf is a leading life science company that develops and sells instruments, consumables, and services for liquid sample and cell handling performed in laboratories around the globe. For more information, please visit eppendorf.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Rafal Grzeskowiak, PhD. Dr. Grzeskowiak is a field application specialist responsible for the Consumables product portfolio at Eppendorf AG headquarters in Hamburg, Germany. Rafal received his PhD at the Georg August University Göttingen, where he investigated regulation of gene expression and cellular signaling pathways involved in diabetes type 2. Later, he took a position of a postdoctoral research fellow at the Max Planck Institute for Molecular Genetics in Berlin and led projects on functional genomics of human dilated cardiomyopathy. I will now turn it over to Dr. Grzeskowiak for his presentation.
All right, Judy. Thanks a lot for this introduction. And hello, everyone, from my side as well, and welcome to our today's webinar, Good Plastics, Bad Plastics, How Tubes, Plates, and Pipette Tips Can Affect Your Experiments. And well, I have to admit, I'm really delighted being here today and bring you this educational and, from my perspective, very interesting webinar. I believe the topic we will talk about here is still not very well recognized and often underestimated in the scientific community. But as you will see, it can have a tremendous significance to the daily routine in the laboratory. So let's move to the agenda for today's seminar. And first, we will generally talk about factors influencing assays in the lab. Then in the second major part of this seminar, I will provide you some review on different classes of leachables and their relevance in the lab. And finally, I will provide you some practical recommendations on how to minimize the influence of leachables and summarize the seminar at the end. So let's move to the first part. Typical factors influencing experiments or assays in the laboratory may be either specific and have direct influence or general, and those have indirect influence on the experiments. And this is what you see here on this slide. Let's consider first the assay-specific factors here presented on the left side of the graph. And typically, these are the factors most researchers or those who work in the lab are worried with and try to control. And these include, first of all, samples, meaning their quality, quantity, integrity, and availability. Then, of course, come reagents, their quality and performance. And, of course, lab equipment with its features and quality as well. Last but not least, great influence on assay performance has the preparation and handling of the samples. On the other side, the general factors believed to have rather indirect influence are often not commonly controlled in the lab routine. And these general factors are very heterogeneous and may include personal skills, lab temperature, regulations, and many others. One of the factors, however, which is commonly underestimated and still potentially very important are laboratory consumables. And this is exactly one of the important topics of this webinar, the importance of consumables in the daily lab routine. So when we typically regard the consumables and their importance in the lab workflows, it is often confined just to the obvious features which are presented on the left, like lid tightness, purity grades, or centrifugation stability. These are by no means important factors and have important impact on the performance on your experiments. There are, however, other, less commonly perceived as important factors, which, as you will see later, may have even more profound effects as commonly believed. And these factors are quality of consumables and very closely related one, leachables, the substances that may leach out of consumables and influence the experiments. And basically, this is the scope of this seminar, of this webinar. So let's recapitulate here the main messages or goals of this seminar. I would like to draw your attention to the importance of consumables and leachables as crucial factor influencing your experiments. Also, I will provide you some comprehensive information on different leachable classes and which classes are critical, and provide some examples, some specific examples on how these substances can influence various assay systems. And last but not least, as mentioned already, I would like you to provide some practical tips and recommendations on how to assess and reduce the risk of affecting your experiments through leachables. Before, however, we proceed to the next chapter, let us do our first poll and ask you some questions. I give over to Judy now.
So the question is, do you regard consumables as important factors influencing your experiments? And your choices are yes, no, or don't know. Give you a minute to answer. Okay, so 73.1% of respondents said yes, 7.7% said no, and 19.2% said don't know. Thank you for participating.
All right, Judy, and thank you for participating. It is really quite, well, for us, good news. I mean, the awareness is there. 73.1% is quite high score, so it's good to hear that most of the participants regard consumables as important factor influencing experiments. It's very good news for us, and yet we'll of course provide you some more detailed information why is so. So let's move to the next chapter, leachables and their relevance in the lab. And let me provide you first of all some important definitions. What are leachables, and closely related and often mixed up term called extractables. So leachables are essentially all chemical substances which can be released from a given container or consumable into the sample or stored material under typical conditions, which include experiment-specific temperature, time, and solvent type. So example here might be, for instance, leaching from a container to a certain drug preparation or your reaction under standard storage or experimental conditions. It means, in other words, and this is important to stress here, that leaching may vary, and sometimes very much, according to these experimental conditions. And depending on these conditions, different chemicals and their amounts or concentrations may be present in the sample and have, of course, in turn, different effects on your experiment. So this is very important to stress here. There are very different leachables compositions and concentrations dependent on these conditions. On the other hand, the extractables are all chemical substances that can be maximally extracted from a given consumable under extreme conditions. So this is basically the worst case scenario. And typically these conditions include very aggressive organic solvents, high temperatures, and long incubation times. So after explaining these definitions, let's move and talk about leachables and their influence in various areas of life. And first of all, this is, well, still relatively underestimated fact actually. Leachables from various household items and personal care products can affect our health. And in various, many clinical studies performed all over the world, it has been confirmed that many leachable classes, particularly two of those, phthalates and bisphenol A, so BPA, are implicated in various health disorders in humans. And well, living in the modern world means, unfortunately, we are constantly exposed to various plastics and leachables from food and beverage containers, different tubings, carpets, cosmetics, and so on. And high concentration of leachables have been, unfortunately, very often reported in general population, and their influence may be very pleiotropic, very versatile, including reduced fertility and reproductive tract abnormalities, also hormonal, brain, and behavioral disorders, but also they are very often implicated in immune functions and various cancers. Now, as you see here, leachables are also environmentally relevant pollutants. And phthalates, bisphenol A, and other leachables have been shown to act as so-called endocrine disruptors and directly target nuclear receptor transcription factors, and by doing so disarray various intracellular signaling pathways with developmental and physiological effects. And these conditions are particularly profound in amphibians, and some of these the examples are shown here. And the scale of the problem is indeed immense. For instance, at least 8 billion pounds of BPA is being used in industry per year and partially released into the natural environment. And of course, as a reaction to this, several countries started monitoring phthalates and BPA contents in various products, and Canada, as the first country, actually declared BPA as a toxic substance. Let's move on. So, how about the lab? How about leachables in the lab? Are they relevant in the laboratory? And the answer is yes, very much. And as you see on the slide, it's actually a very famous science publication from Andrew Holt, where he summarized his research and actually pretty much shocked the scientific community by showing that substantial amounts of leachables are detectable in common laboratory plasticware, and they can significantly affect and hamper actually many assay systems. What he really showed is that, in fact, the leachables pose a likely source of error in many assay systems. So this was kind of a shocking finding to the scientific community. And after this publication, a real flood of various papers have been published, often in high-impact journals, where scientists from different laboratories all over the world were reporting growing evidence for consumables affecting their experiments. And this, in turn, kind of revolutionized the scientific community and led to the conclusion that consumables and leachables were largely underestimated by the researchers and in the past, and pointed, of course, to high importance of high-quality consumables as important factor influencing various laboratory workflows. And of course, this continues. We see here significance in clinical diagnostics. And as you might imagine, when assays go wrong in the research, well, it's pretty annoying. But if this happens in diagnostics, well, it gains a completely new dimension of significance. It might mean the difference between health and disease, sometimes the difference between life and death. And here is one dramatic example published in Nature News from a medical center in Jerusalem, which was routinely screening patients for some rare metabolic syndrome. And all of a sudden, all of their patients, all of the subjects were basically diagnosed as sick and positive, and this appeared actually very weird since the disease is very rare. So it was actually a real epidemic. Of course, they stopped and investigated their routine assay methods, and after a month of investigations, it turned out that problems were caused actually by pipette tips they recently purchased and used for sample preparation. As later revealed, this effect was caused by a dye-solubilizing agent, NP10, used during production process of the tips. So as you see, leachables may have also profound significance in clinical diagnostics. Before we proceed to the next slide, we would like to ask you again your opinion and know some more information. So it's a time for a new poll. Judy, I give over to you.
The question is, how familiar are you with the topic of leachables? Your choices are very, partially, or not at all. Another moment to respond. Okay, so 5.7% of you are very familiar, 71.4% are partially familiar, and 22.9% are not at all familiar with the topic of leachables. Thank you for participating again.
All right, thank you from my side as well. Well, it indicates quite high awareness of the topic. I mean, the majority of you is partially or very aware of the topic of leachables, so it's really high score actually. Well, that's perfect. We are really the audience for this for today's seminar. So thank you again for taking part in the poll. Let's continue. I just close the poll windows. And so let's have now some more details, some specific examples on how actually leachables can affect various bioassay systems in the laboratory. And here is one showing how leachables from a regular micro test tubes can affect enzymatic assays. In the panel on the left, you see bars representing enzyme activity in the presence of DMSO, which was only briefly rinsed through glass control or micro test tubes, here named S-tube. And for information, glass is virtually free of organic substances and typically regarded as negative control in such studies. And as you see here in this example, samples incubated in plastic tubes show up to 40% inhibition of this specific enzymatic activity. And there is also a little story to these figures, which I would like to tell you now. Well, this Andrew Holt, who published this data in a GIT lab journal, he is a pharmacologist and was studying some promising drug candidates and very much interested in their inhibitory effects on some enzymatic systems. And after years of research, he found a very promising compound which showed to his delight a considerably high inhibition rates up to 40–50%. This effect was, however, very unstable and upon closer investigation appeared to be basically related to the tubes he had in his lab, or more specifically a slip agent being released into his samples, namely the oleamide. And as you might imagine, he was very much frustrated, and it was actually the very reason he started as a scientist seriously investigating leachables and their relevance on bioassays. So the conclusion here, it's a specific example, enzyme activity can be significantly influenced by leaching of bioactive substances from the lab microtubes. Let's continue. So the second example shown here investigates effects of leachables on a receptor binding assay, which is one of the most commonly used assay systems in pharmacology and drug discovery. And in this particular example here, the preparations of rat brain receptors were incubated with their specific ligand, and ethanol washes from various laboratory tubes. So in the panel on the left side, you see a percentage of specific binding of the drug to its receptor and the influence of different tubes, namely leachables from different suppliers. And as you can see, they can diminish drug binding up to 50% as compared to glass control. Well, notably, Eppendorf tubes show identical results as the glass control. So the conclusion in this example is that leachables may affect receptor binding assays. And this is another specific example for you, which I would like to quote. And these were very, perhaps, specific assays, so receptor binding or enzymatic activities. But not only such assays can be affected, also very simple, also very routine assays, as in this case, spectrophotometric DNA quantification can be hampered by substances leaching from the lab tubes. And as the title says here, it is really DNA you're measuring? Well, not always. The data shown here originates from a Biotechniques article published by Lewis and his group, and provides quite a striking evidence of some very mysterious nucleic acid molecules appearing in pure water samples upon short incubation. What you see in panel A on the left side is a perfect DNA spectrum with a distinct peak at 260 nanometers, which is merely a heterogeneous mixture of UV-absorbing leachables from the lab tubes used to incubate the water. And leaching process is temperature dependent, as we mentioned already, and as depicted in panel B in the middle, indicating that extended incubations at higher temperatures will lead to more leaching and absorbance. And finally, as shown in panel C on the right, leaching may lead to kind of false DNA amplification phenomenon, which means increase in DNA concentrations after heating the sample, as might be the case, for example, in case of PCR reactions. So this is another specific example. Let's move on. These false positive DNA measurements have been actually investigated in more detail by our application team at Eppendorf. And we actually found this assay a very quick and easy test indicative of general leachable levels and consumable quality in this respect. So it's basically a very quick test you could perform in your laboratory. In our experiments, we did absorbance measurements of water incubated in different tubes for 30 minutes at 95 degrees and calculated theoretical DNA concentrations resulting from the absorbance at 260 nanometers. And the results shown in the panel here are quite striking. Even after relatively short incubation times, the amount of false DNA was reaching over 2 microgram per milliliter. These are the red numbers over the bars, as you see. These concentrations are actually considered as pretty high and sufficient for most assays where DNA is used as a template. Well, of course, in this case, we didn't do any downstream assays, as it surely might be just frustrating because there is no DNA. And the samples showed also high readings at 280 nanometer, which is typically used to quantify protein, and in this case also leading to false positive quantification. So let me conclude here. UV-absorbing leachables can falsify spectrophotometric measurements of nucleic acids and protein. And how about the plates? And the answer is it might get even worse. So here we used the same photometric assay and evaluated absorption spectra of water samples incubated in 96 deep well plates, and the resulting spectra are shown here and represent pooled samples of 3 independent plates. So it's important to note here is pooled samples from 3 independent plates. And most of the plates from different manufacturers tested showed high levels of UV absorbing leachables, and this was pretty much consistent with our findings done with the tubes. What was, however, very surprising is shown in the next slide. And in this graph, the broad red line represents a variance of 96 individual wells of 3 independent plates, and is indeed very high. And this actually indicates that leaching process per se shows very high variability and potentially may falsify or increase variance in assays performed in plate format. So it literally means that dependent on the position of the well, different levels of leaching may occur, and therefore different effects on your assay may take place. It might be of tremendous importance for those who work with different layouts, dilution series, or similar plate position dependent assays. So this is actually quite another dimension of the problematics. First, we have the leachables, then they affect the experiments, and third, there is a high variance to the leaching phenomenon. So we presented you here some specific examples of assay systems where leachables have been shown to be essential factor influencing them. And let's finally see what are leachables exactly and which classes are critical. So, Leachables-chemical analysis, and basically, if we made a brief chemical analysis of typical plastic tube or plate we have in the lab, we would see quite an interesting picture. In contrary to common belief, it's made not only of pure polymer or polypropylene or polyethylene. Apart from polymer molecules, laboratory plastic contains various polymerization byproducts like aliphatic hydrocarbons, oligomers, olefin clusters, oxidation products, and many others. But also, more interestingly, it contains various chemicals added during production process, additives, and their hydrolysis products. And this group is actually largely responsible for most of negative effects leachables have on various assay systems. So let's have a closer look at the additives used during the production process, because these are the substances making the problems. And there are many various additives used by the manufacturers during their production process of consumables, mostly to achieve certain properties of the consumable, or even often simply to reduce the production costs. And in the table here are summarized most additives and are grouped according to their potential of negative influence on various assay systems. So first group contains heavy metals used to catalyze the polymerization, and stabilizers and antioxidants to stabilize the polymers and to extend the shelf life of consumables. And finally, of course, we have pigments in case of colored consumables. And these substances in the first group cannot be avoided during the production process. They have to be added simply to make the polymerization possible and make sure consumables remain stable and for a reasonable period of time. So they are regarded as potentially uncritical, and there is not much evidence actually in the literature of their effects on the experiments. I would like, however, to mention that dependent on the experimental conditions and assay methods used, particularly sensitive analytical methods, they also may have certain influence on experiments. So this was the first group. Second group, depicted here in yellow, is potentially critical and contains clarifiers used to increase transparency. So if you have a very clear tube or plate, it's somehow suspicious already. Antistatic agents to decrease electric charge, and quite heterogeneous group of surfactants and detergents to disperse pigments and other substances. And typically, these substances can be avoided in the production, and as mentioned, have been shown to be potentially critical in some assay systems. And last but not least, the really bad guys. In red, you see the additives that have been shown in many publications to leach into the samples and pose a likely source of error in many assay systems. The worst is these substances can be actually easily avoided and are added simply to reduce the production cost or make it faster. For example, flip agents are used to remove the consumable from the injection mold. And basically, it's the same as you make a pancake. So you put a butter on the frying pan, and basically to remove it faster. Of course, it makes it faster, but the fat is staying on the pancake. So this is exactly the same here. The injection mold is basically the slip agents are used to release the consumable from injection mold faster, but that they remain, of course, in the plastic and contaminate the samples. Also, the biocides or simply antibiotics are critical and are added to prevent bacterial growth of the consumables, on the consumables, which might happen if the production conditions are not very clean. And finally, the plasticizers, they're actually the classical leachables and serve mostly to make the consumable less rigid and prevent breaking. So again, all of the substances shown in red and yellow can be typically avoided in the production process of consumables and are either potentially or critical to most of the assay systems. And as you see here in this small literature digest, these substances are indeed in most classical papers dealing with leachables and have been shown to make sometimes life of scientists really miserable. So what you see here are articles where effects of leachables from different consumable types are shown on various experiment or assay systems. So enzymatic assays, receptor binding cell growth, and spectrophotometric quantifications. And since you will be provided this full presentation as PDF, I will not go into detail of each of these papers, but I think it's worth to read at least some of them later. So of course you will be provided the whole presentation as PDF. Actually, you can access it on the downloads on the website later on. So let's summarize and make some important message in this seminar. There are, as you have seen, 3 critical classes of substances used during production process of consumables that may leach into the samples and negatively influence your experiments. And these are slip agents, biocides, and plasticizers. And if I were to choose just one take-home message from this whole seminar, it will definitely this one. So make sure you remember it. Well, now I think it's enough of scaring you and showing the experiments spoiled by leachables. And here I would like to give you some practical advices in the next chapter, how basically to achieve it and minimize the effect of leachables on your experiments. Before we proceed, however, let's make me another poll and ask you a short question regarding your experience with problems in the lab during the consumables. So I give over to Judy now.
So the question is, did you experience problems in your lab routine due to consumables? And your choices are often, sometimes, never, or don't know. So 6.5% of respondents say often, 45.2% say sometimes, 6.5% say never, and 41.9% say don't know. So thank you again for responding to this poll.
All right, thanks again. Well, let's say, again, from our perspective, interesting results. So it indicates really that most of you actually have problems, and these are related to consumables. Some of you 6.5% even very often. So I hope this seminar will basically provide you vital information how to deal with this. And this is actually the next chapter we're going to now. Thanks again for participation in the poll. And so let's go to the final point of this seminar, minimizing influence of leachables, practical recommendations. And first of all, when we talk about practical recommendations, I would like to remind you one of the first slides in the seminar where the assay-specific and general factors influencing experiments were discussed. And the general conditions in the lab do include consumables and leachables and have to be considered and controlled. What is important here is that this common mindset has to be changed, where only reagents and equipment influence your assay. It is also, and this has to be kind of, this is the message here, it is also consumables and other related factors like leachables that may have sometimes tremendous impact on your experiments. And here are some specific steps how to control and minimize effects of leachables. First of all, it is advisable to do inventory of consumables you use in the lab. Very often, we're not even aware of what we all have and what and why we use for a given experiment. So an inventory will give a good overview of your situation and actually make it easier to react when problems really occur. And very often they will occur, but typically we don't regard consumables as the cause. So in the case of false or variable or irreproducible results, it is always wise to consider possible influence of leachables on your experiments and test respectively reagents, water, and consumables you use. Standardization is also another very good way to minimize effects of leachables. So using same consumables, even same production lots, will definitely decrease the variance in their influence on your workflows. And finally, good laboratory practice is to order new consumables early enough and always test them against all batch in the same experimental setup. So these are basic rules here and basic recommendations for you how to start with dealing with the problem of leachables in the laboratory. Now, another extremely important point to understand is that despite superficial similarity, consumables are not created equal. And although tubes, plates, and tips may appear same to you, they actually significantly differ in material, manufacturing, purity grades, and their performance. And these differences occur in the long process where consumables get their production specification, are produced, and finally quality control takes place, as shown in the upper part of the slide. So the important message here is the right choice of consumables has an impact on the outcome of your assays. And so actually one of the most important practical recommendations here on how to minimize the influence of leachables is to choose right consumables. And firstly, there are many companies manufacturing consumables with various technologies and production standards. It is therefore essential to choose those manufacturers who comply with high international quality and purity criteria and are respectively certified. And there are some important questions you definitely should ask and get positively confirmed before choosing consumable from a given manufacturer. And this is what you see in the slide. In the development and production of consumables according to international standards, ISO norm certification is important, and particularly ISO-conform clean room conditions and high level of production automation is essential. And another important question to ask is detailed information on quality and purity of raw material used for production should definitely not be recycled. And what are exact production conditions and whether any additives are used and which norms are fulfilled. Let's continue. And one very important point which alone would definitely help you minimizing risk of leachables in your lab, is to choose consumables which are free of critical additives. So ask the manufacturer if their consumables are strictly free of slip agents, bioassays, and plasticizers. And well, telling is one thing, and providing in certificates is another. So it's a very important thing here to ask for a respective certificate. Also, for sensitive assays and sensitive analytical methods like HPLC or MALDI, when possible, choose clear consumables to eliminate the influence of pigments and accompanying substances. And finally, it's always good practice to ask for lot-specific certificates regarding consumables quality and purity grades. Important here is the certificate should be issued by external accredited laboratory and should be of course available online, which is not always the case. So these are basically some specific recommendations from our side how to choose good quality consumables for the experiments. And of course, last but not least, it's really important to get professional support in case of problems. It's comprehensive and application sales and application assistance from the manufacturer side is actually very important in case of difficulties with your consumables and respective application. It will save you time, money, and of course a lot of stress. And these were some practical tips and recommendations on how to minimize influence of leachables, and I hope it will really help you avoiding major problems and basically improving your laboratory workflows. And finally, what do recommend top scientists when it comes to leachables? Well, I have here a small citation from Andrew Holt's article in GIT Lab Journal. And he also is an expert in the field of leachables, a leachable guru, as one might say, clearly recommends purchasing consumables from manufacturers that do not use additives and basically avoid suppliers that refuse to confirm the absence of such chemicals. And one of such manufacturers, Dr. Hall recommends, after quite laborious search at Eppendorf, believe me, we did not pay for this statement. It's basically his independent opinion as a scientist. Now, as one of the last slides, one of my personal recommendations in case you would like to obtain more information on leachables and their significance in the laboratory, visit our very interesting, from my perspective, very interesting website at eppendorf.com/leachables, where we place various information and additional resources on leachables, many scientific articles, posters, and other scientific data, as well as, of course, relevant Eppendorf certification regarding quality and leachables. And last but not least, a very interesting interview with Andrew Holt himself, where he talks about leachables and his continuing research on leachables. And we are slowly actually coming to the end of the webinar, so let me summarize the key points we covered here. I really hope to make it clear this point and make you realize that despite common belief, consumables do indeed play a vital role in the laboratory workflow. And I also hope this seminar provided you a good review on various additives used during production process and how these chemicals, by leaching into the samples, may hamper many assay systems. As an important point, please remember the three classes of additives which are critical and shown in the literature to affect experiments, so slip agents, plasticizers, and biocides. And finally, I hope it's also clear choosing high-quality consumables free of these critical additives can significantly reduce the risk of negative influence on the experiments. And I'd like to end this webinar with this little cartoon from a German, pretty well-known journal named Laborjournal, which shows how a lab microtest tube doesn't want to cooperate So cooperate and says to the scientist, well, translated from German, well, if you have to live with this if you take the cheap material. And this, apart from teaching you some German, of course, I think reflects pretty well current developments and change in the awareness of scientific community regarding consumables and leachables and their significance in the laboratory. And from my side, thank you very much for your attention. Before we continue, we have the last poll for you. I give over to Judy again.
And so the question is, was this webinar informative for you? Yes, partially, or no? Wait just another moment. Okay, 90.3% of you said yes, the webinar was informative, partially 9.7%, and no one said that it was not informative. So, very good.
Thank you very much. Well, that's a really nice result. So I'm really glad to hear that it was interesting for you. And we are ready now for some questions. This is one of the good points of this kind of live sessions, that basically after you can ask your specific questions and we'll try our best to answer them. So let's go to Q&A session.
Thank you for that informative. Oh, may I thank you for that informative presentation. Just before we get started on the question and answer session, I would just like to remind the audience how to submit questions. So they can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window, and we'll try to answer as many questions as we can. And the first one is, what are exactly the UV-absorbing leachables chemically? Rafal, should I repeat that one?
Sure. If you would read the first question, I think, what are exactly the UV-absorbing leachables chemically, right?
Yes.
So generally speaking, most organic compounds actually do absorb, to some extent at least, UV light, and highest absorption levels show usually aromatic hydrocarbons, so phenyl groups and double bonds, and typically these are numerous additives with phenyl groups, for example, phenolic antioxidants, stabilizers, clarifiers, and their oxidation products. So these are basically, in most cases, the UV-absorbing leachables.
Are leachables only relevant for assays with incubations in higher temperatures?
Yeah, well, indeed. Leaching is a temperature-dependent process, and generally speaking, the higher the temperature, the more leaching occurs. There is, however, extensive scientific evidence that many critical additives, like SIP agents, bioassays, for example, can leach already and strongly influence various experiments in lower temperatures already. Also in room temperature. So example includes various enzymatic assays, receptor binding, and many others. In the end, it is difficult to predict which assay to which extent will be influenced by a given additive. I think the best practice would be here to avoid the critical additives completely.
Most of the data you show refers to tubes and plates. What about tips? Might they also have some influence on the experiments?
Yeah, although the contact time of samples with the material of the consumables is indeed shorter in case of pipette tips than it is in tubes or plates, they may also have an influence on the experiments. And some examples were provided in the presentation, actually. So arucamide is a slip agent from pipette tips and has been shown to inhibit G protein coupled receptor assays, or dye-solubilizing agent inhibited mitochondrial chain and respiratory chain enzymes. This is this diagnostic case which has been shown, and very sensitive analytical methods like HPLC or MALDI also may be affected by leaching from tips. And there are many more examples in the literature. In general, the more sensitive the assay or the method, the more likely leaching from pipette tip may influence it.
Okay. This next one is a two-parter. Does Eppendorf use critical additives during production of their consumables, and can a respective certificate be obtained?
Yeah, during the production process of Eppendorf does not use any additives, which have been shown in the literature to be critical. So we don't use any slip agents, biocides, and plasticizers. It refers to all consumables, so all tubes, plates, and tips. And we indeed do provide a respective certificate of quality. We call it COQ, so Certificate of Quality, which is of course available online and in each product package.
Can I use the UV leaching test with water incubation to check the quality of tubes I use in my lab?
Yeah, well, in this test, only one class of leachables is actually evaluated, the UV absorbing leachables, which are important, but not the only one, which has been shown to be critical. So there are other classes which will not be visible in this test and still may be present in the given consumable or an influence your assay. So low levels of UV absorbing leachables do not necessarily mean that they are absolutely safe. Also, the conditions of the test and measurement of the spectra has to be optimal so that the results are reliable, and using diluted samples or too short incubation may lead to low signals and basically unreliable readings. So I would say the take-home message here would be the UV leaching test may be done for some brief evaluation, but I would still be cautious over absolute statements, and there might be consumables giving low absorption and still actually having high risk of hampering your experiments.
Is the type of plastic, is the type of plastic relevant, PS or PP, etc.? Yeah,
well, the PS is polystyrene, PP is polypropylene, and they're completely chemically speaking, different plastics, and there are different sets of polymerization byproducts which you will see, and also different additives used during the production process. So definitely, it's relevant. And from my perspective, the PS is much less resistant to higher temperatures. So theoretically, it basically might leach more than PP.
Why do you recommend clear plastics? Well,
it's simply because it's colored consumables, colored plastics have some additional more additives regarding exactly the color. So there are the detergents, there are the substances dispersing this dyes, and basically most of the dyes also can be found later on as a leachable in the sample. So whenever possible, I would really, for sensitive methods, analytical methods, would try to work just with the clear consumables.
Okay. In your slide 19, why are ssDNA 25 nucleotides incubated 30 minutes at 95 degrees centigrade? As we know, PCR heats the samples only 5 minutes at 95 degrees centigrade. Let me know if
well, this is the experiment. This is basically conditions which are published, and this is actually to better visualize and basically make this more clear. You have to remember that PCR takes place and the samples are heated for 5 minutes by this repetitive process. So in the end, it's basically you have, let's say, 25, 30 steps, and each of those includes 95 temperature or 60 for elongation, and basically it's a cumulative process. So in the end, you will have a very similar profile. We tested this, and basically we did a normal PCR or a standard incubation at 30 minutes, 95 degrees, and the results are quite similar.
The next person thanks you very much for your informative talk and says they actually have the other side of the problem, with samples being especially low concentration ones getting absorbed by plastics. Do you plan to have another talk to cover this aspect?
Yeah, well, it's a pretty well-known phenomena. The biomolecules, proteins, nucleic acids bind to surface of a plastic, typically polypropylene. And it's basically due to certain hydrophobic domains which these molecules contain. And upon binding, of course, first of all, you have less material, and second, there is certain denaturation and a lot of activity. And we have certain class of consumables, so-called low binds, which basically addresses this problem. And if there is, of course, interest regarding this topic, we're willing to provide such webinar. So thanks for the information, and I think it's a very interesting topic, actually. So why not doing another webinar on this? Thank you.
The next one is there a good way to test our consumables for the presence of these agents besides testing in our assay, which may be hard to run? Chemical tests, absorbance?
Yeah, as mentioned, so one of these very simple tests might be performing this UV absorbing test, which is basically incubating water and doing the spectrophotometric measurement. This is not covering all of the leachables. So as I mentioned already, it might give you some indication, but I would be still cautious. The best, of course, would be HPLC, but this is costly and very complex method. In the end, the best way, and this is one of the recommendations from this webinar, is to be on the safe side and basically purchase consumables from manufacturers which do not use critical additives.
Okay. The next person says, are you going to repeat this type of presentation in the future? Since this is my first time to attend it live, I will just add that this presentation will be on demand for 6 months. I don't know, Rafal, if you want to answer further.
Well, yeah, this is actually what we do. We provide this presentation on demand, and on top, if you would like to have a PDF of the presentation, will be available on the microsite as a download. If you have any questions, of course, my email is provided there, so basically you can address any questions writing directly email to me. And yeah, I would remain with this actually here.
Okay. The next person thanks you again and says, I have a question of whether you perform some experiments/assays about leaching of additives, etc., before marketing your products. And do you think we must ask our suppliers about such assays before we purchase plasticware?
Yes, we do perform routinely quality tests, and well, first of all, we start with specification, product specification, and of course, for all of the consumables, there is no critical additive. But yet, we do test general leaching of all of the organic substances, so we routinely perform such tests. And I do recommend that you ask your supplier about how basically they address this topic, if they test it, if they certify it, and what is basically their statement on this.
How does leachables affect samples or assays when stored at lower temperatures?
How do leachables affect? Yeah, as I said, it's a very complex process actually. It's a diffusion, there are some chemical reactions taking place, but in the end, there are different classes of different substances going in different temperatures and times into the sample. So basically, the key message here is, basically, if you avoid consumables with critical additives, you're on the safe side. But to answer the question how basically they affect is very difficult. It really depends on the exact conditions in the experiment. So basically, the solvent you're using, the temperature, and the time are critical factors here. And if you're interested in particular assays, so I imagine your assay, basically we're willing to provide you the literature and where you can basically search through and find if there is any information on the assay. It's very difficult to say if for a particular assay and it's given temperature there is any effect of the leachables.
Okay, the next person says, just wondering about the manufacturers that were compared during the internal lab tests at Eppendorf. How stable are the Corning consumables in comparison with Eppendorf with respect to leachables, especially the culture flask?
Yeah, well, this is an educational webinar. We will not provide any specific information on the competition. This is not our policy here. But of course, if you're interested, these part of these results are published in scientific journals. And we would like, if you're interested, we provide you the articles here. But we, as a company, as a manufacturer, we don't provide names on competitors or details on competitor comparisons here.
Okay, here's another one for you. What about the effect of leachables on long-term storage conditions? Yeah,
most of these papers of this data is regarding short-term effects, but I might imagine and the time factor plays a huge role here, and the longer the time, the more leachables you have. And in most cases, also the temperature takes place, so it's kind of tricky to predict. In my opinion, there is just one paper, I think, regarding this so far. In generally speaking, we don't have much data on the long-term storage, so it's still kind of under evaluation, and I think there will be coming scientific papers on this. So at the moment, we don't have much data on this.
Okay, and why are those two wavelengths used, 260 and 280, in order to measure leachables?
Again, as you might remember, we were showing the whole spectrum, and 260 is basically the prominent peak where most of these UV-absorbing leachables absorb, and actually it's a peak where DNA absorbs as well. So this is the wavelength which we chose to basically quantify the false concentrations of a given molecule, in this case DNA. And 280 is protein. It's typically used to quantify protein. So to answer shortly the question, 260 was chosen to simply say how much false DNA you would have, and 280 how much false protein you would have.
Okay. How can you be sure that an experimental result is influenced by consumables or leachables and not by other factors?
Yeah, this is a good question. It's always, there can be many things actually. So it can be the equipment, can be reagents, can be the PhD student. And the best way is actually, if you have an experiment and you suspect there is something wrong with it, just do the same experiment with different consumables. And one of the consumables should be a good quality, so without critical additives. Of course, the best way would be to control with the glass, but I'm pretty sure it's not always feasible. So use glass as a negative control in your experiments would be the best option. If not, choose the high-quality consumables which are free of critical additives.
Okay. It looks like we may have run out of time. I would once again like to thank Dr. Grzeskowiak for his presentation. Do you have any final comments, Rafal?
Well, from my perspective, it was a very good webinar, and judging from the information we got from colleagues, the awareness is very high, which is very positive. And I enjoyed very much, and I wish to all of the participants good luck with their experiments, and thank you for the attention.
Thank you. I would also like to thank our sponsor, Eppendorf, for making today's educational webcast possible. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. Today's webcast will be available for on-demand viewing through April 26, 2016. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. FAQs and the presentation's PDFs will be available for download after the webinar in the resources section of the microsite. See you next time. Goodbye.